Hi, my name is Stephannie Seng, and I'm a Master's of Science candidate at San Francisco State University in the Connor Lab. My studies are in cell molecular biology, and I merged this with my current research in ecology and evolutionary biology. My current thesis is titled Drooling over Insect-Induced Galls, Salivary Origins of Exogenous Cytokinins. So what are plant galls? Plant galls are abnormal growth of plant tissue caused by another organism. From viruses to invertebrates, there are a variety of organisms that can cause these tumorous growths to develop in plants. The mechanism of gall induction is known for some gall inducers, such as in bacteria, but it is completely unknown in insects. Our research aims to answer questions related to how insects can cause these abnormal growths of plant tissue. The picture on the left shows a picture of our plant gall, in which our fly, shown on the right, feeds in the larval stage and further develops into the adult stage before emerging. Cytokinin is an important plant hormone. It contributes to a variety of plant processes, such as cell growth and differentiation, the formation of roots and shoots, and the redirection and transportation of nutrients. The insect may be using cytokinins to reallocate sugars and nutrients to the site of where the fly pierces the plant. The intention may be gall induction or sustenance for the larval insect. This site is known as a mobilizing sink, where nutrients and sugars are highly concentrated. This plant growth hormone is also found in gall-inducing insects in active and inactive forms in very high concentrations, up to 10 times the amount in plants. My project has 3 specific aims. The first, to identify genes involved in cytokinin production in the insect Eurosta solidaginis. The second is to determine if cytokinin pathway genes are overexpressed in salivary glands relative to body tissues. And lastly, to determine which insect pathways are responsible for cytokinin production. My project encompasses a wide variety of techniques and methods. These include field work, in which we collect specimen in Minnesota, greenhouse work, involving fly rearing and gall induction. We also utilize wet lab work, in which RNA extraction and reverse transcription are used, digital droplet PCR and antibody staining. We also use microscopy for the microdissection of larvae and confocal microscopy. And lastly, also bioinformatic and computational work involving DNA sequence analysis and primer design. In conclusion, the overexpression of genes in salivary glands compared to body tissue would suggest cytokinins are being synthesized in the salivary glands of the insect. Such a result would be the strongest evidence yet that gall-inducing insects are producing cytokinins to manipulate their host plants. Lastly, I would like to thank my PI, Dr. Edward Connor, Gabriela Ponce, and the Connor Lab. And also, I would like to thank my funding sources as well. Thank you.